walk a little bit?
Actually, I don't like that.
Ataxia is a gait disturbance that is caused by the inability to coordinate the sensory and motor systems used in walking. Observe that this patient walks with her legs far apart, giving her a wide base. Despite this wide base, she is still awkward and unstable.
It's pretty clear, you don't see
A
smooth.
tandem walk forces the patient to contend with a narrow base while
You
walking.
don't see smooth movement.
Note the tendency to fall as this patient attempts a tandem walk.
What's kind of interesting here too is, you've probably heard of police stops. They suspect you drinking. They might have you do a walk like this. And because alcohol also impairs motor coordination, right? That if you talk, like I used to work with disabled people, and I worked with a guy who'd been launched through the front window of his car, who survived as a teenager. He ended up with massive cerebellar damage. And when he walked like he was drunk. The numerous occasions the cops accosted him and accused him of public drunkenness, when in fact it was brain damage to the cerebellum, had nothing to do with alcohol. But easy to mistake that. You saw that, right? Easy to mistake that for maybe drunk walking. So you can see why the cops maybe were suspicious or something. But so that's just kind of classic motor discoordination that you see with cerebellar damage. But what you see with cerebellar damage kind of depends on which part of the cerebellum's damaged, which makes sense, right? It's not homogeneous. Just like with the brain, the hemispheres, if you damage visual cortex, you'd expect to see visual deficits, blindness, scotomas, things like that. If you damage temporal lobe, you probably expect to see auditory language dysfunction. So same with cerebellum, it's not homogeneous. So let me show you some other things you can see.
The principal signs of cerebellar disease are dysmetria, dysdiadochokinesia, and ataxia.
So we just saw ataxia, right? That's the term for kind of discoordinated movement. Dysmetria, metric, dysfunctional, refers to problems like with the force, and the directional, the direction of your movements. If you could all do this, like try and take your finger and touch your nose, right? Can you do that? And did any of you bash your finger in your nose? No. But if you couldn't adjust the force, you might actually launch your finger into your nose and give yourself a bloody nose. Or if you couldn't adjust the direction of that limb, you might launch your finger not into your nose but into your eyeball, and then you just destroyed your eyeball. That would be problematic. So the cerebellum is required for that. So if you look at, again, it kind of depends on which part of the cerebellum is damaged, but you can see things like this. So let's look. Here's
Dysmetria is the inability to coordinate a complex motor activity involving several muscle groups.
So here they're.
In this example, the patient is asked to repeatedly touch the examiner's finger and then her own
You can
nose.
see they're overshooting.
Note that the initial trajectory is misdirected and that the ensuing mid-course correction overcorrects and the finger misses the target.
So again, think about how problematic that would be if you're just trying to do something as simple as touch your nose or scratch your nose. But if you initially, even if you eventually get to the nose, but you initially overshoot, you can drive your finger into your eyeball and blind yourself. So that's a problem. So this is dysmetria. Then one of my favorite words in neurology, dysdiadochokinesia, means problems like, especially with kind of rapid sequence movements, especially alternating movements. And like I used to play this with my grandma. You ever play this game where like one person puts their hand out and you have to put your hand on top of it and then they go like, you ever do that when you were a little kid? I did that with my grandma. That's rapid alternating movement. That kind of thing gets decomposed and people have problems with any kind of rapid alternating movement or snapping your fingers quickly or whatever it is, right, left, right, left, right, left, or imagine coordinating two hands to play the piano or something, I don't know. So that's dysdiadochokinesia. And do we have it on this one? might not be so apparent for you when you look at this, but, so she's being asked to just do rapid movements, and, you know, it does appear like she can do it. I mean, she can, but, often with patients, it's slower than normal, and it's not as smooth or rapid alternation. So anyways, enough movies. And so, you know, think about what we saw Tuesday. These are different phenomena, right? I showed you videos of Parkinson's disease, classic vasoganglion disease Tuesday, where we saw slowness of movement and people have problems like getting up out of a chair. Let's also compare all of this with this, Unit 2. Because Unit 2, we talked about motor systems in general and a lot about cortical spinal tract, the major motor system in the brain. So with that, what did we see? Paralysis, weakness, right? Okay. It's not that the patients with cortical spinal tract lesions have movement that is no longer smooth, is jerky, it's just that they can't move, okay? That is not what you see with cerebellar damage. You don't see paralysis or weakness, so it's right there that's showing us it's involved in kind of a different aspect of motor control. Do you know what I mean? But so let me show you an example of a cortical spinal tract lesion patient, which I should have shown you in Unit 2. Also, do you remember we made this distinction between upper and lower motor neuron disease, right? Do you remember if you damage an upper motor neuron, a CNS motor neuron, we could see enhanced reflexes. So let me show you an example of that. And usually, you know, you got to compare left and right because I guess I can't show you that. Here we go. Right-sided hyperreflexia. You got to compare right and left because often, right, an upper motor neuron lesion is because of a stroke on one side of the brain. So you only expect to see weakness, paralysis, or changes in reflexes on the other side. You see what I'm saying? Not on both sides of the body. So you got to compare, if you're doing reflex testing, both sides of the body, just to know like what's normal, what's baseline. But here, let's look at, if you were wondering. So they're doing a patellar reflex test, right? They're tapping the patellar ligament, and then here, see, that's what we mean by hyperreflexia. It's maybe not blowing your mind, like, you know, that's it. But compare left, pretty minor, to right. Okay, so right side of hyperreflexia. Where do you think the damage was? It was probably on the left side, right? Because the left side of the brain controls the right side of the body. Anyways, that's kind of stuff that you see with upper motor neuron disease, cortical spinal tract lesions. She probably has weakness, paralysis too. So that's just totally different from cerebellar problems. No weakness, no paralysis. We're not talking about that kind of stuff, okay? All right. So kind of cool thing about cerebellum is okay, the first amazing thing is, you know, let me draw a picture of a really bad picture of the brain. So this is like the hemisphere from the side. That kind of looks like a sperm whale, but whatever. This is the front, this is the back, and then here's the cerebellum back here, and here's the brainstem. Spinal cord. Temporal lobe, frontal lobe, parietal lobe, occipital lobe. Okay, so simple prototype, standard issue, you know, Walmart human brain. Okay, so here's the cerebellum. So one thing is, you know, that's a pretty accurate size representation. It's pretty small, right, compared to the hemispheres. Guess which thing has more neurons? This thing in blue, which we call the cerebral hemispheres, or this thing in red, which we call the cerebellum. The cerebellum actually does. So it's like super small, and yet it has as many or maybe more neurons as the rest of the brain itself, which is kind of mind-blowing, okay? So its ability to do complex computation is just off the charts. So that's the first thing. Then if you conceptualize the cerebellum as like a box, okay? It's getting, okay, so there's the box, that's the cerebellum. So, here's your motor cortex. This is like hiding conceptual, right? Okay. It's another box, okay? And our motor cortex output via the cortical spinal tract goes to muscles and we can move, that's cool, right? That process the other side, cortical spinal tract, that influences like muscles, okay? But it turns out, when we send information down to the spinal cord and the cortical spinal tract, we send the same information to the cerebellum, okay? So the cerebellum is getting informed about what we're trying to do. What's the motor we're trying to do? Got it? Okay. Now, what do we call those little cool little sensory receptors in muscles that sense like changing length? Muscle spindles, right? So that stuff, muscle spindle, you know, there's also 3-neuron pathway or whatever. That's going back to primary somatosensory cortex, okay, and it's telling the brain about like, did my muscles stretch? We also have Golgi tendon organs, right, which is telling the brain like how much tension is on my muscles, right? Okay, but guess what? That information is also going to the cerebellum. Okay, so the cerebellum is getting information about sensory information about like as we're doing a movement, what's the angle of my joints and how stretchy are my muscles and what's the tension under my muscles, and it's getting information from motor cortex about like what's my intention, what am I trying to do with movement, and so it can compare what we're trying to do with actually what happened in real time. How does it know what happened in real time? Because like, right, the angles of my joints and the tension of my muscles, and so it can compute then an error signal, you know, again, how actually it does this is like, you know, go into a neural lab and, you know, figure that out for us and publish it, but it can compare, it can come up with an error function for like what we wanted to do and what actually happened, and then over time, and then it can feed back this error information to cortex, and so then cortex can make adjustments, and then, right, over time we learn movement. Does that make sense? That's pretty cool that it can do it. So that, I mean, you know, and you could imagine if it can't do that, why you would end up with discoordinated movement, okay? If you can't do that kind of error correction. By the way, I think I've said this previously, one of the problems with robotic firms making robots, okay, that move around the earth, is they can make really cool robots that move fast and even that jump and can, but they don't have sophisticated sensory proprioceptive feedback yet, okay? And so the robot's movements often seem clumsy. It's almost like they have cerebellar damage, and yet they don't have the cerebellum. But so you see, this somatosensory information is so important for feeding back to the cerebellum so we can adjust error, right? So that's kind of big picture. Let's now get into it. So that's a great question. A lot of what we see with babies falling and stuff is they don't have fully myelinated cortical spinal tract yet, you know, the major motor system. And since it's not fully myelinated, it's not fully functional. And since it's not fully functional, they have problems with movement. And likely too, I don't know exactly when cerebellum fully myelinates, but there's likely that's an issue too. Yeah. You know the frontal lobe, you know, we're gonna finish today motor. Finally, we're gonna get into like higher cortical function, which I hope you find interesting. But you know the frontal lobes, which up till now, we've only thought about them as this motor control center. But what do we really think about when we think about frontal lobes? Like higher thought, personality, planning. You know, when they finish myelinating, like at age 25, some people joke that maybe they never fully myelinate, like guys. So, I've heard people make jokes like that. But anyways, so the myelination of different parts of the nervous system takes place at different times, and so early on, that cortical spinal tract is not fully myelinated. Okay. So. So, remember how there was a basal ganglia loop? Cortex talks to basal ganglia. I'm going to do it now. See. Basal ganglia talks to thalamus. Thalamus talks back to cortex. There's a cerebellar loop too. Cortex informs the cerebellum about what we're trying to do. Cerebellum talks to thalamus. Thalamus talks back to cortex. So there's a cerebellar motor loop too. Okay. And you know, both of these loops are influencing ultimately what those motor tracks do that we talked about in Unit 2, all right, which is, you know, all this stuff down here. This big line would be the major motor tract, cortical spinal tract, you know, whatever. Can I just say one other thing? We now know, probably from the last 20 years, cerebellum isn't just involved in motor control. I'm actually not going to talk about it, but new research says it's involved in language and maybe even in emotional processing, all kinds of cognitive functions. So like the old view, in any textbook you pick up probably still has the old view. Cerebellum, motor coordination, end of story. No, that's not the end of the story. And if you think about it, it makes sense. The cerebellum, if it has as many neurons as the hemispheres of the brain, it's probably involved in more things than just motor coordination. So if you get into it, if you're interested in the cerebellum, you'll find out it does all kinds of interesting stuff. You could imagine why it might be involved in language. Like there's a timing function there too, like stringing words together and sequencing them, right? Anyways, another interesting thing about the cerebellum that's different from cortical spinal tract. Let's review. Left side of the brain, cortical spinal tract that starts in the left side of the brain, remember, controls the right side of the body. That's not how the cerebellum is set up. Right side of the cerebellum controls the right side of the body. So, in contrast to a stroke up here in the brain, where if you have a left-sided stroke, the symptoms are on the right side of the body. If you have a left-sided cerebellar stroke, you have symptoms on the left side of the body. Okay, it's not crossed. Got that? So it's ipsilateral relationship, which is interesting. It's just different. Okay, so it's a comparator. This is what I drew. You know, we're telling motor control centers in the brain are telling our alpha motor neurons to, you know, contract a muscle, but we're also sending the same information to the cerebellum, like, hey, this is what I want to do. I want to pick my nose. And then as we're picking our nose, we got all this proprioceptive feedback that's getting fed into the cerebellum, so the cerebellum can compare. I wanted to do this. This is what actually happened. There was an error. Let's deal with that error. Motor cortex. And so cerebellum sends the output of that error computation to motor cortex, and motor cortex adjusts. How cool is that? Okay. Sometimes the cerebellum is called the silent zone of the brain, because this is interesting. You know, you think back to previous units I talked about. If you're doing brain surgery, let me draw a bad brain again. Okay. Here's my little sperm whale brain. Okay. There's the cerebellum. That's so bad. Okay. And then here's the central sulcus. So remember, primary motor cortex in front, primary somatosensory sensory cortex in back. So if I take an electrode during neurosurgery and I stimulate motor cortex on a human, or a primate, or a dog, any mammal, I'm gonna see discrete movements in different parts of the body. And it depends on where I stimulate that cortex, right? Because there's a topographic map, right? Okay. If I stimulate somatosensory cortex with an electrode, I'm gonna see, the patient will tell me, because we do neurosurgery typically on awake patients, they'll tell me that, they'll tell us that they feel something in my finger, in my toe, okay? So if you did the same experiment with the cerebellum on a patient during neurosurgery, you took an electrode and you stimulated the cerebellum, you'd think you'd see movement since it's playing such a role in motor coordination. Most of the time you see nothing. Then you might think, okay, well maybe they're gonna feel something. Right? You're applying like electrical current to the cerebellum, they don't feel anything either. So that's why sometimes it's called the silent zone of the brain. That doesn't mean, like, if you damage it, you're not going to see major problems. I just showed you video evidence of that. It just means, like, if you stimulate it, for whatever reason, you don't really see much, which is kind of interesting. So, more big picture. Why we should care, what actually happens. So, yeah, this is so bad. People are starting to leave. You guys can bring fruit and tomatoes and shit and throw it out in your room like the rest of school. That's it. You know, let's spice things up. Instead of leaving, right? Let's just make it more interactive. Just throw shit out. So, anyways, you've heard of Pavlov conditioning. Did I just use a bad word? Sorry. You've heard of Pavlov conditioning, where if you take an animal and you present it with food, it will salivate. Makes sense. Like I salivate if I see sushi in front of me, or mac and cheese, or a hamburger. Okay. If you then tap a tuning fork, nothing happens, because typically, you know, humans and dogs don't eat tuning forks. Now, stage 3 of the experiment, if you present the dog with really tasty doggo treats and simultaneously tap a tuning fork, the dog will salivate. Not because of the tuning fork, because it's got its favorite kibble food there. If you do this enough time, you've all heard this, right, from intro psych. If you do this enough time, now if you just tap the tuning fork to that trained, conditioned dog, it'll salivate. So it's learned to associate the sound made by the tuning fork with the fact that it's about to get a really great meal. You know what I mean? Guess what? That requires the cerebellum. If you damage the cerebellum in this experimental dog and you do the same training protocol, it will not make that connection. Okay. Here's another one. I've always wanted to get like these prisms. I've read about them, but any of you ever go to a bar where they have like a dartboard and you throw darts in? Apparently now, the new thing is like they have axe bars, which is like, what could go wrong? Throw axes at something in the bar. Anywho, imagine if you're wearing prisms where they shift, you think you're looking straight ahead, but they shift your view by 15 degrees. Okay. So now you go to that dartboard. So, you know, dartboard's over here, I'm wearing these weird prisms. I think I'm gonna throw the dart straight at the dartboard, but it's probably gonna veer 15 degrees away because the prisms are distorting my view, you know what I mean? Guess what? Over time, we learn to adjust. We learn to compensate. And we can hit that, you know, if you're good at darts, we can hit that dartboard eventually, even though we got these crazy prisms on, okay? Guess what? If you have cerebellar damage, you won't learn to make that adjustment. That motor learning will not happen. Here's another example. We talked about this in Unit 2. You know, it's so important to maintain visual fixation on something, right? For obvious reasons. And if I'm walking and I'm trying to look straight ahead, it may not seem like it, but my head's moving a little bit, but I'm trying to maintain fixation. So it'd be great to have this reflex that, like, if my head moves a little bit to the left, my eyes move to the right, you know what I mean? So I got that fixation. That's called the vestibular ocular reflex. Here it is being demonstrated on this woman. And guess what? You need a cerebellum for that. So there's also input to the cerebellum from cranial nerve 8, the vestibular ocular, you know, I'm sorry, the auditory vestibular nerve. And so another thing we see with cerebellar dysfunction is problems like with coordinating head and eyeball movement and stuff like that, okay? And also you can end up with like dizziness, vertigo. Do you guys know what you call rapid alternating eye movements? Nystagmus. So you can see all those with cerebellar disease too, okay? So those are just kind of interesting things. Oh, one last one that I always thought was kind of cool. You know, you've heard about people going to like the space station, and some people are up there for months, and they're adjusting to zero gravity over time. When they come back to Earth, it's a heck of a time, like just walking, because it's like they haven't been in gravity for a long time, and so they have to readjust to Earth gravitational field, etc. That requires the cerebellum, we believe. Okay, so just a teeny bit of anatomy. This is a brain, and we just cut it in half, okay? This is the mid-sagittal view. And see my point, see how teeny the cerebellum is compared to the hemispheres, and yet there's as many neurons here as here. Other than that, in some ways it's similar architecture. There's gray matter on the outside, and then there's white matter on the inside, okay? One difference is, remember in the cerebral cortex, there's typically 6 layers. Cerebellum, there's 3. And early anatomists, when they look at like the cerebellum in cross-section, they thought it looked like a tree. To me, it looks kind of like cauliflower or something, but they thought it looked like a tree. And here's a ventral view, like a bottom view of the brain. And you can see there's 2 cerebellum hemispheres, just like there's 2 cerebral hemispheres. And they kind of are on either side of the brain stem, okay? So that's just kind of showing you a little bit of anatomy. This isn't an anatomy course. And here too, if you took a different cross-section through the cerebellum, this is a mid-sagittal, this is like a transverse, you'll find out there's actually some deep nuclei in the cerebellum. Remember, we've got some deep nuclei in the brain too, called them the basal ganglia. We've got some deep nuclei in the cerebellum. I don't want you to memorize all their names, but I want you to know that here's another kind of big picture thing. If this box is the cerebellum, okay, we've got lots of input coming in here from different places, like proprioceptors, like cranial nerve 8, you know, what's the head doing? Is it moving from cortex, right, motor information from cortex, whatever. And there's all kinds of crazy, complex computation going on. And then eventually the cerebellum has to output, right? It has to talk to the rest of the nervous system. The only part of the cerebellum that can output are the deep nuclei. So there's this box inside the cerebellum box is like the deep nuclei. These are where the neurons are that talk back to CNS. Does that make sense? So you got all these inputs coming in there, crazy computation, error correction, you know, adjust timing, adjust force, adjust the sequencing of movement, and then if we want to output, it's only the deep nuclei that can do it. Okay. Now, if you look at a whole cerebellum, this is what we're looking at here, okay? There's actually 3 main parts. So there's a center vermis. There's a little floccular nodular lobe, and then we have this massive kind of lateral region of posterior lobe, okay? And if you damage the vermis, that's where you see that kind of ataxia, that drunken walk sort of phenomena that I showed you in that video, okay? Right. If you damage the floccular nodular lobe, that's where you see things like nystagmus, dizziness, vertigo, problems with the vestibular ocular reflex. So this is kind of like the part of the cerebellum connected to cranial nerve 8. And if you damage the lateral part of the cerebellum, this is where you see more complex stuff, like problems with rapid alternating movement, dystatokinesia, with like tiny force of movement, right? So there's functional, anatomical, segregation, specialization, you know what I mean, in the cerebellum. It's not all homogeneous, okay? Right. So again, 3 parts. The follicular nodular lobe, because it's getting information from cranial nerve 8, we can call that the vestibulocerebellum. Remember, cranial nerve 8 had an auditory part and a vestibular part. Okay. The vermis, that's the part of the cerebellum getting all that input from, like, the dorsal column, medial lemniscus tract, you know, with all of that muscle spindle information and Golgi tendon organ information, right? So we'll call it the spinocerebellum. And then this massive lateral part of the cerebellum, you know, people think that's kind of like more evolutionarily evolved or whatever. This is involved in more complex motor control, timing, sequencing of movements, right? You know, if you're wondering, like, what do you mean sequencing of movements? Or just think about like a quarterback throwing a football or someone doing salsa dancing, you know what I mean? It's like complex sequence of movements. If you're trying to play a sequence of notes on a guitar or whatever, like, all that has to be timed just right. So that's kind of lateral cerebellum, okay? And here too, maybe put a little note. I don't want you to, you know, worry about the names of all these little deep nuclei, but just the point here is there's a lot of places of the nervous system inputting into the cerebellum. It makes sense too. If it has as many neurons as like the whole brain, as the hemispheres of the brain, it's probably getting massive input. So it's getting, again, input about what our joints are doing, what's the state of our muscles, right? Spinal inputs. It's getting information from cortex, especially like what is our movement motor planning right now? What am I trying to do movement-wise? Trying to, whatever, pitch. Okay. And it's getting input from cranial nerve 8, like what's my head doing? Is it spinning? You know, is it rotating? Okay, all right. And then it's going to output to all kinds of different places. Okay. Again, and just one example. I tried to really just eliminate all the anatomy, all the details, since that's not what this class is about. Just one example of an input. So this is, hopefully, this seems kind of familiar. We got proprioceptors, and remember their cell bodies are in the dorsal ganglion. I actually asked you to, I guess I asked you to draw the spinal thalamic tract. But anyways, on your exam. And so this information makes its way into the spinal cord, and a lot of it makes its way up to the brain, right? And we're conscious of like, hey, where's my body in space? But some of it, instead of going up to the brain, it just goes right into the cerebellum. All right, so just wanted you to see that we have one example of an input pathway in the cerebellum. Okay, all right. One neat thing in the cerebellum is, here are those homunculi again. Remember the homunculus? Okay. Map of the body. We saw this on primary motor cortex, primary somatosensory cortex, right? We saw a map of the retina and visual cortex. We saw a frequency map in auditory cortex. And here we have body maps again in cerebellum. And it makes sense if it's like controlling movement or at least sequencing movement and stuff like that, that we have maps of the body. You know what I mean? Okay. So we got topographic maps in the cerebellum too. Just a theme of this course, like how the brain's organized. Okay.